In this lecture, we're going to begin Chapter 9, which is looking at the taxonomy of bacteria. And so this first lecture will look at taxonomy and look at one of the early taxonomist, who is a guy named Carl Linné, who changed his name to Carolus Linnaeus. And then we'll talk a little bit about how biologists identify species when they find something new that they've not seen before. So taxonomy is the science of classifying organisms into groups based nowadays on evolutionary relatedness. So we look to cluster together into further and further refined groups organisms that are most closely related to each other. And standardized names are essential in biology because there's no other way. If we don't have a standard name for an organism, then we don't have a way to talk about that organism and know exactly what we're dealing with. So that term taxonomy comes from it's either Latin or Greek, I'm not sure which, but the idea of a taxon, which is a category into which an organism can be grouped. And so the goal or the work of taxonomy is first to establish the criteria that are going to be used to identify those organisms, and then based on those criteria, we cluster together organisms that have the most characteristics in common with each other. And the secondary goal, so one is to get everything organized so that we can kind of have a systematic way of dealing with all of the diversity of organisms that we find around it. And then a second goal is to provide information about how the organisms evolved. So to build within this taxonomic organization scheme information about evolutionary relationships as well. And so you heard me just a second ago use another term, systematics. Systematics and taxonomy, those are the same idea. So a systematics just means a systematic way of organizing and classifying organisms. So what we developed then is a group of taxa in which the higher level taxa, we see fewer and fewer characteristics that are shared, that are in common. So the larger, let's try this again. The larger, the higher the grouping, then the more individuals that are found in that grouping and the less they have in common. And as we work our way through the taxonomic system, we weed out into other groups those that are least like the organism that we're most interested in, so that we winnow down. We essentially make a funnel from up here at domain, which you've heard about, until we get down to an individual species. So many organisms are found in this grouping domain, and when we get down to the very end of our taxonomic system, then we've got a species where there's only one type of organism is found in that cluster. Okay, so where did all this start? It started in the 1700s with a Swedish physician whose name was Carl Linné, and he is generally credited as the founder of the science of taxonomy. And so he established a system of what's referred to as binomial nomenclature. Nomenclature just means naming. So this is naming. Binomial means two names. So it's a two-name naming system. And he developed it using some of the names were crafted in a way that they reflect Latin, the way that Latin is written and said. So within this binomial nomenclature system, all organisms have two names. So for instance, the human the human species and or genus name, and the specific epithet that follows is Homo sapiens. So Homo is the genus name, the specific epithet or the species name, the specific epithet is the second term there, is sapiens. And so currently there are no other living Homo genuses, no more species within the genus Homo. But as we look back in the fossil record, we can find a number of species that had that genus name Homo. And so collectively, together, the genus name and the specific epithet is referred to as the species name, or sometimes you'll hear people talk about the scientific name. So species name and scientific name are the same thing. And he also established the system of classification based on shared characteristics. So now, he did not include domain. Domain is a relatively recent addition to this, but our classification system looks like this. At the highest level, the most inclusive group is the domain, and then domains are divided into kingdoms. So for instance, within eukarya, we've already talked, there are 4 kingdoms plus a whole bunch of kingdoms that are being worked out. So we have fungi, fungi, plants, animals, kingdom Animalia, kingdom Plantae, kingdom Fungi, and then all of the Protist kingdoms. I should have said 3 kingdoms plus all of the Protist kingdoms that are being worked out. So domains are divided into kingdoms. Kingdoms are divided into phyla or divisions, depending on which group you're working with. So the plant people tend to talk about divisions rather than phyla. Phyla is what the animal folk tend to talk about. Within bacteria, we'll be talking about phyla. Phyla can be divided into subphyla, and all of these have sub and subgroupings within them. As we're looking at classification of humans, which is what this classification scheme here is showing, there's humans are in kingdom Animalia. They belong in the chordate phylum, and within the chordates there is a subgroup called the subphylum Vertebrata. Then following phylum is the class Mammalia, so all of the mammals. That includes cows and pigs and cats and dogs and monkeys and apes, and go on and on. All the grazing animals in Africa and so on. Classes are divided into orders. Humans belong in the order primates. Orders are divided into families. So the family Hominidae is the family that humans belong to. And then families are divided into genus, and the plural for genus is genera. So you may see me use that term, genera. So genus, and then there's the specific epithet that's attached to a species. And I do want you to know this classification system of domain, kingdom, phylum, class, order, family, genus, and species. And a good way to remember that is with this mnemonic here. Dear King Philip came over for good sex. I'm sure I didn't say that out loud or even write that on a PowerPoint slide. Okay, so just, I want you to know this classification system of domain, kingdom, phylum, class, order, family, genus, and species. And I want you to know which one is going to be the most inclusive, have the most species that are in common. That would be domain and kingdom. And which one is going to have a grouping that has the fewest shared characteristics among all the individuals. That would also be the domain or kingdom that is going to have the greatest number of species and the least in common with each other. whereas we get down to families and genera, and those are going to have the fewest numbers of species with them, and they're all going to have the most in common with each other. Okay, hopefully that makes sense. So, generally, that's a bad, I gotta fix that. Generally, individuals in the same species aren't divided into smaller subgroups, but we do have, especially here in the bacteria, we see exceptions to this rule because we talk about strains of bacteria. And a strain of a bacterium is a culture that has grown up from a single organism. So, for instance, if we had a petri dish and we had done some plating and done this and then pull out here, then when we set that to incubate, what we might see is colonies here where we have spread out. So you have a really dense spread here, lots of growth. We now pull from there and then sterilize the loop, pull into here, sterilize the loop, so that when we get this last pull here, hopefully we've got just a few individual colonies that we're spreading around. And so if we were to take a sample from this colony here that had grown up from one bacterium, that would be a strain of the bacterium. And strains can have different characteristics from each other. So this strain may have one set of have just by chance have a slightly different genome than the strain that's right next door to them. So strains exhibit small but permanent genetic differences. It could be around the need for specific nutrients, the resistance to certain antibiotics. This is why we have Staph aureus that is not particularly harmful, and then because we have antibiotics that we can treat it with, and then there's MRSA, there's the methicillin-resistant Staph aureus groups. So this is where we see that kind of individual characteristics. So generally we identify different strains by a name or a set of letters and numbers that follow the specific epithet. So for instance, Clostridium difficile, C. diff, if you will, these are 3 different strains. There are more than just these 3 strains, but these are 3 strains and some of the nomenclature that's used to distinguish between the 3 strains. Okay, so nomenclature, again, that's the naming of species, and if we use binomial nomenclature, then there's a genus name followed by a specific epithet. How do we write this down? So the genus name is always capitalized. So for instance, here we see Clostridium difficile. The Clostridium is always capitalized. Escherichia coli, the E is always capitalized there. The specific epithet is never capitalized, so lowercase specific epithets. And if you are writing it by hand, so if you're going to write Clostridium botulinum, which is another species of Clostridium, then you would always underline it. So Clostridium botulinum. Sorry, those extra little lines there weren't intended. The keyboard or my tablet just didn't recognize it. So after that, after you've written it down once, you can later refer to it as C. botulinum. Again, underlined if you're going to refer to it this way. So first reference, you always write it out. A later reference in a paper or in something that you're writing about it, you can just abbreviate it as C. botulinum. What else to say about it? If you are typing this up, then you don't have to underline. You can just do what I did here, which is make it italics. So italics equals underlining. So underline if you're doing it by hand, though, because I don't know if anybody who can write in italics. Okay, and so let me turn the page here, make sure that we're on it. So what are the characteristics then that are used to classify things? Well, Linneaus was working primarily with physical characteristics, and so Linneaus didn't have all of the tools that we have now to classify and to systematically organize the living world into neat little boxes. His work, he did it mostly on appearance, and early microbiology was also done primarily on appearance, things that you could look at as you're looking at the organisms under the microscope. So things like cell shape, or how the membrane took up stains, or how structures in the membrane took up stains, excuse me, how the organisms moved, that sort of thing. But as systematics has matured, as this discipline has matured, the goal has shifted to also include information in the classification scheme that tells us a little bit about how closely organisms are related to each other, and a little bit about the evolutionary history of the groups that are being studied. And where we get a lot of that information now is thanks to molecular biology and some of the techniques that molecular biologists can use, including sequencing the nucleotide bases that are found in the genome of bacteria. And so with the advent of molecular biology, so work that has been done over the past 30, 40 years, we have seen a real revolution in taxonomy, as we've talked a little bit about in class. And we can be more precise in understanding evolutionary relationships. And so because of that, there's been a huge, just this huge turning over of all of the classification schemes as we've come to understand the information that molecular biology provides. So what is a species then? And that's a relatively easy question to answer in eukaryotes. A species is just the group of all the individuals across the world that could potentially interbreed and produce young who are also capable of producing their own young. That's what we mean by viable young. So it's a relatively speaking, there are those who would argue with me about that, but relative to bacteria, it's a pretty cut and dried definition of what a species is. If we can get them together, they recognize each other and will mate and produce young, then and the young can also reproduce, then we've got a species, or they are part of the same species. But this definition isn't quite so useful in bacteria, because bacteria don't mate and then exchange genetic material as a part of their reproductive strategy. So we don't see two bacteria mating and then swapping gametes with each other. That's not the way it happens. Bacteria do exchange genetic material, and they do it with other individuals within their species, and they can do it with other individuals across species. And they can even pick up dead, just chunks of DNA that are found in the environment. So the organisms don't have, as we've talked about, the organisms don't have to be living in order to be able to share DNA with another bacterium. So those things make it really messy then, just trying to we don't have that neat cut and dried. If they mate with each other and produce young together, then they must be the same species. So instead, we have to look to a lot of other characteristics. We look at the biochemical reactions that are happening inside the cell. We look at the chemical composition of the molecules inside the cell. We look at cellular structures, at genetic characteristics, and we can even use immunology. So we can use antibodies to help determine whether there are specific proteins available in the membrane or in the capsule of an organism. So we're going to look at each of these as a way of classifying bacteria as we get deeper into the chapter. So last of all, if we find a new organism, how would a biologist go about identifying what species it is? If we find an organism that's new to us, well, we use something that's referred to as a dichotomous key. Dichotomy means separation, and so we use a series of yes and no statements to separate the potential species that it could be from each other, and then make our way through this series of yes-no questions to narrow down the list of potential organisms, and eventually we get down to choosing between two individual species, and a yes-no question is used to distinguish between the two, to allow a positive ID of the species. So for instance, here, oops, wrong way. Here we've got a group of, that's weird. Okay, oop. Sorry about that. Here we've got a group of organisms that we've identified, my pen has stopped working, but we've identified as having a high guanine to cytosine ratio, and they're gram-positive. So we've already done some narrowing down of our bacteria, and we know that it has a high GC ratio and is a gram-positive. So then the next question is, does it form filaments, or are there no filaments? And if the answer is yes, it forms filaments, then we look at, well, actually, let's go this direction. Let's say that no, it doesn't form filaments, this bacteria that we have. So, okay, we've got no filaments, no mycelia, those are the filaments. And then we check to see, is it acid-fast or not? If it's not acid-fast, then we look to see, does it stain gram-positive or gram-negative? Well, it stains gram-positive. Does it cause diphtheria, or does it make propionic acid? If it makes propionic acid, then what we have is Propionum bacterium. If it doesn't make propionic acid and instead causes diphtheria, then it's Corynebacterium. So we've isolated now it down to either one species or one genus or the other. So suddenly my pen is working again. So we went from being a high GC ratio and gram-positive to the fact that it had no filaments, that it wasn't acid-fast, it's gram-positive, and it made propionic acid, and so it's Propionobacterium. Okay, there we have it. Everything you wanted to know about introducing to you the idea of taxonomy within bacteria. You want to know what to do if you have questions. Okay, I'm almost ready. I'll be able to leave in about 3 minutes. I thought I would be. What the heck? Come on. Oh, I'm going to be screaming if I've lost it. Damn God. There we go. Okay. I entered it.